Well, good morning, everyone. We are 1 minute past the hour. Welcome to the first workshop of the Include Data Hub, a very auspicious moment, I would say a historical day for the field of Down syndrome research. My name is Joaquin Espinosa. I have the privilege of serving as the leader of the Administrative and Outreach Core for the Include Data Coordinating Center. I'm also the executive director of the Linda Sunic Institute for Down Syndrome. And to break the ice here, I would invite you, all of you, to introduce yourself through the chat. Maybe just your name, your institution, your interest in Down syndrome, something brief. We won't have time to open up the call for all of us to introduce ourselves, but we can do this trick of introducing ourselves through the chat so we'll get to know each other. My personal objective for today is to get out of the way as soon as possible so that we can get to the juicy part of the workshop this morning. Let my colleagues in the Data Coordinating Center demonstrate to you the use of the Include Data Hub. But just in case some of you are new to the Data Coordinating Center, I'll give you a very brief introduction about the DCC. Hopefully you're all familiar with the NIH INCLUDE Project, investigation of co-occurring conditions across the lifespan to understand Down syndrome. It is perhaps the best thing that has happened to the field of Down syndrome research ever. It's a trans-NIH research initiative established in 2018, focused on improving the lives of people with Down syndrome through advanced biomedical research. It has nearly quadrupled new funding for Down syndrome research since 2018. And this investment is allocated into three major areas: high risk, high reward, basic science approaches, assembly of a large cohort study across the lifespan, which is related to the activities of the Data Coordinating Center, and inclusion of individuals with Down syndrome in clinical trials. Now, why does the name of the project include, talks about the co-occurring conditions of Down syndrome? Trisomy 21, the genetic cause of Down syndrome, causes a different disease spectrum in people with Down syndrome, protecting them from some pathologies and predisposing them to others. Now that there is a sizable population of adults with Down syndrome, and now that we can analyze their medical histories, it has become evident that the extra chromosome somehow protects them from development of most solid malignancies, hypertension, atherosclerosis, allergic sensitization. On the other hand, having an extra copy of chromosome 21 predisposes these individuals to Alzheimer's disease, autoimmune disorders, various leukemias, severe COVID-19, congenital heart disease, autism, sleep disorders, seizure disorders, and other conditions. So it becomes clear that if we study the population with Down syndrome, we'll make discoveries that will not only serve them first and foremost, but hopefully also will make discoveries about the etiology of these medical conditions that are modulated either positively or negatively by the trisomy, that also, of course, affect the general population. With all that being said, it is important to note that there is massive inter-individual variation across individuals with Down syndrome. The individuals pictured here in this slide that may have none of these co-occurring conditions and who may be capable of playing musical instruments, giving inspirational speeches, have a job, go to college, what have you. There may be other individuals in this slide that may be burdened by multiple co-occurring conditions. They may be nonverbal, perhaps with a dual diagnosis of autism, so on and so forth. So it is in this inter-individual variability that then we have an opportunity for discovery, right? We like to say that a precision personalized medicine approach will clearly benefit people with Down syndrome. So what drives this remarkable phenotypic variation? Is it driven by genetic variants, either on genes encoded in chromosome 21 or genes elsewhere in the genome? Is it driven by epigenetic mechanisms? Are these different manifestations of the trisomy correlated with different endotypes that can be detected in the transcript, on the protein, the metabolome, the microbiome, the immune profiles? Furthermore, what are the contributions of the environment, lifestyle, socioeconomic status, cultural factors? Clearly, there are a lot of ideas here. There are a lot of possibilities. So we need an orchestrated effort that would enable the testing of many, many hypotheses quickly and efficiently. So with this in mind, the NIH called for the creation of a data coordinating center to accelerate discovery of the etiology and biological pathways underlying the co-occurring conditions of Down syndrome by facilitating access to an analysis of data from the cohorts of individuals with Down syndrome that are being funded by the INCLUDE project. So the data coordinating center was created and it consists of 3 cores: the administrative and outreach core, providing management and outreach and education. We lead this out of the Syriac Institute here in Colorado. The data management core, tasked with managing data collection, processing, and harmonization, led by our wonderful colleagues at Sage Bionetworks in Seattle. And then the data portal core, tasked with facilitating access to an analysis of data via a web portal led by my colleague Adam Resnick, who's going to speak next. So why would the NIH task the Data Coordinating Center with creating a data hub and portal? Well, because I think it's well demonstrated now that knowledge hubs and data portals are massive accelerators of research across many different scientific disciplines. It's hard to imagine the day when we didn't have things like PubMed or GeneCards or CBio portals or KidFirst or what have you. So today, we are very happy to share with you that the Down syndrome research community has a world-class data resource similar to those developed for the study of other major medical conditions. So here we are today. IncludeDCC.org portal, IncludeDCC.org, the Include Data Hub in beta mode with the first few cohorts integrated. We're gonna spend the next almost 2 hours playing with this amazing resource, and hopefully we'll have a very interactive workshop where you'll be able to understand how to explore the available data sets, clinical profiles, whole genome sequences, targeted proteomics, metabolomics, and let me assure you there's a lot more data in the pipeline arriving to the data hub in the months to come. Now, before we get to the fun part, I would like to do a couple calls to action. If you haven't checked our website, IncludeDCC.org, please do so and subscribe to our email list there so that you can stay updated with any developments from the Data Coordinating Center. Also, if you are an INCLUDE Fund investigator, you have a profile in the website. It's a great place, this investigator registry, for you to find collaborators with similar interests. I'm, for example, pulling out here a screenshot of Philip Lupo's profile, showing the grants and publications associated with the INCLUDE project. So please review the profile and let us know if we got things right. This is a bit of a semi-automated process that leads to the creation of the investigator registry. Let us know if you need any updates. You can always email us down here at info@includedcc.org. Another call to action here. Today we have a workshop, but if you really want to increase literacy in the data science within the INCLUDE project in your research team, we have an opportunity here for you. This summer we'll be running a DS3 training event. DS3 is standing for Data Science for Diverse Scholars in Down Syndrome Research. This is a 2-week immersive course, hands-on, focused on big data handling, analysis and visualization. It is focused on graduate students and postdoctoral fellows from underrepresented minorities. 2 weeks, July 11 to 22, at the University of Colorado, Boulder. Wonderful place to be in the summer. All expenses covered, travel, lodging, meals, etc. Applications are due May 25th, so there isn't a whole lot of time. Please promote this with your minority trainees. Take a moment to take a screenshot here. tinyurl.com, T21 data. It's a very simple application. We want to train your diverse scholars in the big data science of Down syndrome research. And I believe that is my last slide. No, I have one more. How can you participate? How can you help the field and the Data Coordinating Center? Well, first and foremost, continue doing great research in benefit of people with Down syndrome. When the time comes, please collaborate with the DCC team when approach about integrating your cohort and data sets into the data hub. As you will learn today, we have a virtual biorepository. You have an opportunity to promote your biobank, your samples through the virtual biorepository. And I want to call your attention to some funding mechanisms. There are many funding mechanisms by the INCLUDE project, but two that are very related to the activities of the Data Coordinating Center, the EXO-1 funding to generate data from your samples, and also RO3 funding for data curation, harmonization, analysis, and sharing with INCLUDE DCC. And also prepare for a data intensive future. There's going to be a lot of data arriving to the data hub in the months to come and beyond. You can always stay updated of the funding opportunities by the INCLUDE project at this URL here, INCLUDE project funding. And that is my last slide. And with that, I will pass the podium to my colleague, Adam Resnick, and I will be monitoring the chat. I'll shut up now and serve as moderator. Please, as Adam and then the other colleagues start demonstrating the hub, feel free to enter your questions in the chat. I'll be monitoring those and I will be addressing those in real time. Adam, all yours.
All right, thanks so much, Joaquin, for the great introduction. And as Joaquin highlighted, really the work here at the Data Coordinating Center spans a number of teams across the nation and in Canada and elsewhere, really coming together to really innovate on how it is that we empower data sets and discovery across the community and across modalities. So I'll do a brief introduction and then I'll turn over to JP, Justine, and Jack at Seven Bridges to really move through some of the portal demonstration and analysis within this framework. One thing that I'd like to highlight is that, the portal and its launch is really represents a starting point. So in the same way that data sets will continue to come in, our teams are going to continue to innovate and add features, development, initiatives, and iterations around the data hub and its implementation. Next slide. All right, so the introduction, I always like to sort of frame, what it is that we're trying to accomplish through the deployment of an infrastructure like a data hub. And really, it's to try and bridge across the diverse activities that are required to drive the scientific process, beginning with samples or clinical or phenotypic or other data that are typically collected by diverse community members across the research and stakeholder community, to transform that those samples and the initial data that's generated from them into really harmonized information that is queryable and can be analyzed across those data sets as if it was one large data set in ways that can drive new knowledge creation and then enhance the impact on behalf of participants and or patients. And that process, historically has often taken shape in siloed environments in ways that are serially connected across very long timelines. And much of the hope is that through the building of new infrastructure and the deployment of the data hub, we can actually accelerate this process and enhance the connectivity and use of the data sets and the stakeholder community members across platforms and resources. Next slide. So for the initial launch, our priorities were really twofold. The first is to support a framework for cohort building through enhanced cross-data findability and accessibility across an initial set of cohorts that really serve as the launch point for the data hub. And as we support that cohort building activity, pair that activity with the deployment of a cloud-based computational framework that can support harmonization, shared use of resources, and standards for the analysis of that data in really a facile and accelerated manner. Next slide. One of the key challenges we currently face and will continue to face as we build these types of portal and resources is that in order to maximize the discovery potential in the setting of complex data sets, we really actually have a diverse community. We have community stakeholders who, as they interact with the technology space of data science, on the one extreme are people who are very used to coding and working at the command line interface around data. And then at the other extreme, we have users who are really working at the knowledge and informational context, who need facile tools to explore and point and click across the data in ways that support the hypothesis generation and or validation. And you actually want both extremes and everybody in between to be co-resident in one platform. And that's really our objective. And across this spectrum, you'll see a number of logos that really represent some of the technologies that we'll be continuously implementing within that framework. Next slide. And across this sort of a technological framework, you'll also, if you go one slide back, JP, across the technological framework, you'll also see diverse community stakeholders. So you'll have researchers who are all the way at the command line side, and you'll have researchers who are all the way at the portal knowledge base side, and you'll have clinician and even stakeholder community members who can essentially fit along that spectrum. But each one of those stakeholders may actually have different types of questions that they want to answer. So it's really this combination of dimensionality across the types of questions that you want to answer and the tools that you have at your disposal to answer those questions and to collaborate across that framework that is one of our goals. Next slide. Now, Include Data Hub is doing a number of new things. And some of the exciting components of the Include Data Hub is that it's a true hub that begins to collect data resources across multiple different sources in ways that, as far as I know, is really some of the most diverse data source context being brought together. So for example, you'll see in the demonstration that one data source is DS Connect, and DS Connect represents data essentially generated or submitted by stakeholder community members and participants themselves. Historically, most of that type of data is typically isolated and discrete from data generated by researchers or academics. In the Include Data Hub, we're going to bring those types of data together with data sets generated by researchers. And that research community actually, even already at launch, represents research data sets generated by an institutional setting or consortia setting, and then research data set that are sponsored by the NIH across several different programs. It's really, that's, the second or third, depending on where I started, enumerating challenge that we face is really integrating across these sources of data and the kind of data generation that are typically generated differentially in that setting on behalf of that diverse community and diverse expertise setting. Next slide. And so with the launch, I just want to take a moment to highlight a couple of firsts that essentially are already taking shape. And what that means is that, we're really, as a team, extremely proud of this initial launch and tackling some of these first, but also recognize that whenever we engage in first, there's always room for improvement. So the Include portal, at least for our team, and I think more broadly, is a FHIR first enabled infrastructure. I won't go into the details, but FHIR is really a technology that standardizes the use of clinical and phenotypic data within a really facile environment for application program interface and use of that data. As I mentioned, there's already the integration of participant provided data and investigator generated data, as well as linkage, at least to our knowledge, for some of the first times, into the biospecimens and the biorepository context. Again, bringing what are typically siloed frameworks of use within the research context, and most of the times, biorepositories and associated limb systems or data with those biospecimens are typically segregated from the derived data or the analytics of that data. And here we're bringing that to bear in a common set of environments. We've also iterated on the accessibility of the data and essentially navigating both the required security and privacy framework that respect participants' need for privacy, while at the same time empowering discoverability by developing a registered tier framework for authentication and use of these data. And that authentication is really an emerging standard that we're continuously developing across both the INCLUDE DCC, but also other partners and frameworks. I mentioned the biorepository, and I'll turn it over now to JP to really go through the data exploration tool and the merges across this cohort building activity, a file repository, and a virtual biorepository. And again, this really just serves as an initial webinar and touch point with you as a community in the hopes that you will provide iterative feedback in ways that we're very eager to hear, both positive and negative, to inform the optimization of these resources. All yours, JP.
Thank you, Adam. And before JP takes us on the journey here, I would like to remind everybody, this is intended to be interactive. We won't be able to unmute everybody and have lively discussions. Maybe we will towards the end, but we are monitoring the chat. If you face an inconvenience, if you have any questions or comments, please enter them in the chat. And while JP is demonstrated, we'll be taking care of those. JP, all yours.
Thank you, Joaquin. Good afternoon, everyone. My name is Jean-Philippe Thibert. I work with the St. Justine team, with the INCLUDE portal development team in particular. And so my role with the team is I'm the product owner / analyst as well. So as most of you know, today we're going to be presenting the portal, and we'll be presenting essentially the features that we have for it. And we, as Joaquin and Adam mentioned, we really want to welcome any type of feedback or questions or comments that you all have, because as I mentioned, this is really going to be an iterative process, as much from the data ingest side. So we're going to be having new data sets over time, but we're also going to be implementing new data features. So your feedback is really what's important, and we want to make this portal useful for all of you in terms of research, even if you're a community member or clinicians. So for the portal demo, we have here again the URL. This is the official INCLUDE portal URL. We're going to be going over 3 of the main sections. So we're going to be going over the user registration and authentication. So sign up and register via Google and ORCID. We'll be going over the dashboard to show you a little how to connect to the platforms for data access and analysis. And finally, the big piece here will be the data exploration. So to browse data and build cohorts for data visualization, accessibility analysis, and finally to copy data files into from INCLUDE to Kavatica. So I'll just exit this screen here and I'll open up the portal. So this is the portal. I'll just move, sorry, my little screen here that I had for Zoom, and maybe I'll move this one too. All right, perfect. So you all should still see this. This is what we land on if we provide the portal URL. So this is a landing page, and we at first sight see the available data. So this really encapsulates what we have in the portal at the moment. We currently have 5 studies, 6,000 participants, 27,000 biospecimens, and 55 terabytes of data files. So although we launched last March 21st, we have a good amount of data files already. So it's already a very useful tool. And we do provide here a little bit of a description about the portal. So just to let everyone know what we're all about. And since this, as I was mentioning in terms of feedback, one way that you guys could all do that is by clicking here on the feedback button powered by UserSnap. And this button will always be present in the portal on the right-hand side. So if you do click on that, you could simply click and drag. It'll take a screenshot of what you are looking at. And you can provide your feedback from here, fill in your email and submit, and we'll receive that on our end within our UserSnap account. And we'll take in any type of feedback, especially if there's any errors. We definitely appreciate that. And we'll act on it as quick as possible. All right, so let's get going. We'll all click sign up, and I encourage you all to follow along. Currently, we have 2 authentication services. In the future, we do want to implement the ERA NIH ERA Commons NIH authentication service. However, we're waiting currently on some paperwork, but we should be getting it soon. So let's start with Google. I'll have here one of the accounts. Oops. All right, little glitch there. So this leads you to the click-through agreement and essentially through the terms and conditions. So make sure that you read and understand that. Just click next, and you'll be redirected to a user intake form. So essentially, you'll be filling out your name, your affiliation, your research role, and data use. So let's just go ahead very quickly and fill that out. I'll just put here other for now. All right, tester. Interesting. And this is optional, and I will write here that for my data use, I'll be learning more about Down syndrome and health outcomes. So submit that. And what you'll be redirected here to a customizable and personalized dashboard. So really this dashboard is where you're going to start your exploration of the data. You will be building saved virtual cohorts, saved filters, and down the line, we do expect to include various analytical tools and internal external workspaces. So what we see here is—
Can I comment here on what you just did, where the users, create a profile. And there was a box there where we asked the users to tell us about the research interest. This is a very important exercise to create the community of users that we hope to achieve. As we are showing right now, there's a community link there. So please take a moment when you register to tell us what you're doing, like you have done here in the chat in the introduction. This will be useful for all the users to recognize you, to know that you have shared interests, and perhaps start collaborations or some other type of networking. So it is important that you let us know why you're coming to the portal for and what you hope to achieve, even if you have actual intent for collaboration. What is it that you need? And if Adam, you want to add anything about that piece?
Yeah, let me add just a couple of comments though, as what Joaquin was highlighting. Some of the downstream portal development will be actually the capacity to connect amongst users of the portal and to actually query a framework around common or shared interest within the data landscape. But the other part that I just wanted to highlight is, oftentimes one of the barriers for access and use of data sets is the need to request permission to use that data. And here, what JP and Joaquin are highlighting is what I reference as this registered tier framework, essentially providing a way for us to really display and empower data in ways that meets regulatory requirements by the NIH and the data stakeholders themselves, in order to much more rapidly provide access and query of some of these data, as opposed to more traditional paths of a process of requesting access, review, and then receiving access notification in a serial manner. So really, what we've done is iterated around the form by which we can both empower connectivity and collaboration, while at the same time meeting the regulatory hurdles and frameworks of authentication and use of certain tiers of data. So that's the other reason for collecting this information, is to meet those requirements on behalf of the data use.
Very good points to both of you. Thank you. All right. So in terms of the dashboard here, we have a few things. It does look a little bit empty, but I'll come back to the dashboard as we progress through the presentation. But just to give a little bit of information here. So yeah, as I was mentioning, the dashboard will essentially be where you're going to start off, and you're going to be exploring data and your safe query, your safe filters and sets. What we have here is a few redirects to the studies page and the data exploration page. And we have a few widgets here that are movable and personalizable. Again, to provide a little bit more context, I'll come back to the dashboard as we get to each piece, and I'll show you a little bit more how these widgets can be a little bit more useful for your cohort building activities. Here, I just want to address as well that the Include DCC website team did an amazing job building a help resource, and they're going to be presenting this Monday, the 16th. And so this is really a great tool to walk you through various features of the portal, what type of connections that you need for to the different platforms, data access, really. So it's really a great tool. And in the similar vein, we also have the website, which provides a little bit of an overview about Include as a whole, the studies, and the teams involved in the project, really. So you'll be learning a little bit more on that on Monday if you tune in. And as Adam and Joaquin were mentioning, we also have a community tool here, which essentially gives an overview of all the users that are registered within the portal. And here we display the affiliation and a little bit about the data use of each individuals. Of course, this is a first version. And so as Joaquin and Adam were mentioning, we really want to empower the community, whether it be connecting community members with researchers or researchers connecting with other researchers based on research topics of interest. So we're really going to be building this tool to empower the community. Jumping to the studies page here, this lists the different research data sets that we have. So we currently have 5. We have HTP, DSCOG-all, maybe the names are a little bit more speaking to you all, DS Connect. And so we have the study code, which here, if you click on it, again, will bring you back to the Include website. Yeah, there you go, a little bit of a load. And it will provide a little bit more details on the study itself. Here we have the study name and the program. We have the dbGaP accession number and the number of participants associated to the study, the families, the biospecimens, and the different data types that they have for the particular studies. Now let's get to the important part of this workshop. Let's talk about the data exploration tool that we have here. So really what this tool will be is, as mentioned, will be a driving force, and it's really going to be the workhorse of the portal. It will be meant to empower you all to build virtual cohorts based on the queryable filters that we have here on the left-hand side. So really the use here is if, let's say, you have a research topic of interest, or you have an interest that applies to your specific scenario, whether it be a particular diagnosis, or if, let's say, you have sleep apnea, or you're interested in research on sleep apnea related to Down syndrome, well, then you could use the filters on the left-hand side and build your virtual cohort accordingly. So what we see here is that we have 3 different categories. This is based on the participant, the biospecimens, and the data files. A major upgrade is that from our other portal that we work on is the Kids First portal, is that here we merge the participants, biospecimens, and data files. So the queryability across of these 3 categories are all linked. So let's say here you have 6,000, well, 6,000 participants, and you'll see this a little bit better when I'm going to query across the filters. But these correspond to the 27 biospecimens and data files. So as you'll see, we'll build a few queries here. You'll see that it updates accordingly. So let's go over here on the different types of filters that we have. Each category has their respective filters. So here we could filter it by study code, by their Down syndrome status if they have trisomy 21 or disomy 21, the diagnosis, the phenotypes, sex, race, ethnicity. For the biospecimens, we have the different sample types. So if you're looking in particular for DNA samples, for plasma, etc., you could search through there. The collection sample types, this is really at the highest level. So from the draw of, for example, the drawing of blood of a patient, for example, the age, the availability of the sample. So if it's available, if it's unavailable in a biobank, and the laboratory procedure and biospecimen storage. Here for the data files, we also have the access level. So I'll talk a little bit about that once we get there. We also have the data categories, so genomics, proteomics, metabolomics that are available in the portal, different data types, the experimental strategy, and the file formats here. So these could all be expanded, they could be collapsed, and there are a few things. So let's start by building one query here. And we could, just to show you all how the query builder, so this is what I refer to as the query builder. So let's add a filter here for the study code HTTP. So we now have that. And let's say we want to add for participants that are females. So as you saw, we updated here the number of participants. And now we could see the number of biospecimens corresponding to these participants are 14,000. And similarly for the data files. So another thing that a great thing about the query builder is that you could easily create a new one. So you start off fresh. I'm now back to 6,000 participants. So essentially all the participants in the portal. And I could easily just do a second query. So let's just do the same one, but let's add male here. So now we can compare between the participants in HTTP that are now that are sex male and sex female. And what we see here, whatever is depicted and displayed as blue is what your active query will be. So you could easily switch from one query to the other. So now this will be the active query. If we were interested in combining these, so unfortunately we cannot add, you cannot be a female and a male. So we could combine these two queries with the OR operator. And so we're going to see that we add, essentially we combine these and we see that we have 686 participants combining both. So let's close this. You could also duplicate the filters, delete them here. And again, we see the number of participants here. Another interesting thing that we could do here is we could save a new filter. So if, let's say at a later session, you were really interested by this set of filters, this combination of filters, you could actually save this. So let's say HTTP as the, whoops, sorry about that, as the cohort. And let's write sex, male, female. So that will now be my new filter, and it was saved. And so let's come back to the dashboard. We should now see it here. So essentially the save filters is a way for you to, an easier way for you to use, instead of going across all the filters that you have, especially if they're long. Now I only had 2, but if they're very long, then it's a lot easier to simply save them. And then you click here. You can also adjust the name here directly and delete them directly. But you could click here and then you'll be back with the same query. So let's add here a few things with the save filters is that you could easily add a new filter, and I'll display that right now. Let's actually do one right now. Let's follow the same. Actually, let's come back to the first one. I'll duplicate this filter and I'll add here a diagnosis for it. And this is an interesting filter that I want to show you all. This is what we call the diagnosis browser. We also have the same one for the HPO phenotype HPO browser. And essentially it provides, not only do we have the diagnosis for the specific participants, so this column will show the exact diagnosis with the participants with this exact term, we also show the hierarchy of these diagnoses. So this is following the Mondo ontology. And so you could drill down and find a particular diagnosis, and it'll show you the whole tree. And we could drill down all the way to the bottom here. So as you can see, we could see Down syndrome. And if we're looking here, it means that essentially we have 212 participants, including the descendant term. So this, we have here the higher node. It'll encapsulate all the participants all the way to the bottom node. All right, so we could also search here on the search bar. So if we want to add celiac disease, for example, we'll see that we have 23 participants with this exact term. And we could easily apply this. And if we were interested, we could either use the any of, all of, or none of operators. So let's simply just add this here. And we could also, we could do the same here. Actually, this should be okay. So once you do a modification, you could save the filter, and now it's saved. And if, let's say you have a saved filter and you do some modifications to it. So let's say we go back here. Actually, let's go in HPO. And let's do congenital malformation of great arteries. So if we add that, as you guys saw, there's now a new icon that could discard the changes that were done to the save filter. So you could simply click this and you'll come back to the state that you originally had with your save filter. You could also delete the filter, share a URL of the save filter, and you'll also have a drop down of the different filters and you could easily switch back. So as you saw, this was the original one that I created. So that covers a lot of the features that we have here. We could clear all the queries that we have. We could remove the labels. This is especially useful when you have multiple diagnoses or phenotypes that have longer names, and they also have the ontology codes there. So that could be useful at times. And we also have the operators here and or within the query itself. So we could change that and you'll see that it dynamically updates. So coming down here to the summary view.
JP,
yeah.
JP, there is a question in the chat that is very relevant to what you just demonstrated about filters. And the question is from Rusu about, are these studies still being updated? Will we receive notification when there is an update with the filters we say? So this is a great opportunity for you to explain how the filters work as more data arrives to the portal versus how, you see where I'm going. We have anticipated this type of question.
100%. And this is it's very good that you mentioned it because I forgot to mention it actually. So here within the save filters, to your point, thank you again for the question. To your point, we're going to be receiving a lot more data. And so this is going to affect whatever filters you have. So what we plan on doing is, we haven't done it yet, but we plan on incorporating a type of alert icon. So whenever you have a save filter and it's affected by the data ingest of, if we have a new data set in the portal, then we're planning on having an alert icon here just to let you know in case you expect the same results, but they're actually not the same. So it's important to capture that. But once I get to the save sets, this is maybe a little bit more of a static option in terms of using saved IDs and sets. So I'll touch a little bit on that once I get there. But great question.
In addition to the iconography and notification in your dashboard, the other things we'll pilot, if users choose, is notifications, for example, via email or some other format of alert that, one of your filters results has changed, and you can come back to the portal and learn from it. So I think it's a really great sort of innovative space around this cohort building activity that we're looking forward to iterating on with users.
Definitely, good question. So jumping here to the summary view. So what we see here really is an aggregate, a visualization of aggregate data. So we have various different charts. Here we have the demographics, and you could hover over and you'll see, for the most part, this applies to most of the charts. If you're hovering over, then you'll see the values. And so you'll see across here, here. And if you're interested in one of them, then you could simply click here and it'll be added to your active query. So now I just started a new one. And that'll dynamically update all the other charts as well. So I'll just remove that here or just start a new query. And similar to what I was explaining here with the facets for the Diagnosis Mondo and Phenotype HPO browser, we also have a sunburst that follows the same type of mechanism where essentially you could select a phenotype of interest. And at this point, we're at the highest level, the highest node. And so we could drill down to the lower nodes all the way to the bottom. Let's say here, language impairment, delayed speech and language. And so you'll see here that you'll have the path of the hierarchy. And so you could either click here to go back, or you could also click in on the arrow here to go back. So it displays the number of participants with this particular phenotype, or if it's if it includes the descendant terms as well. So if we were interested in the delayed speech, again, similar to the other charts, we could add the term to the active query and then it'll update accordingly. So I'll just make sure that everything's fresh. And this is a similar situation for the Diagnosis Mondo. You could drill down, similar to the HPO. We also have here the participants by data category, so just displaying the different types. And again, this is a very useful tool for community members, especially, or for anyone really, if there are certain research topics or scenarios that affect your situation. So let's say you're looking into males with this particular diagnosis, then this provides a quick visualization to show what type of data that's available. And you could look a little bit down deeper if you're interested in data analysis. And finally, the last chart here is the participants by data type. And so we could hover and we see that the values really, so somatic structural variations, we have 207, variant calls 228, and so on. So that covers, oh, sorry, I forgot to mention this. And similar to the widgets, you could adjust this, the various cards and charts, and place it to your liking. And essentially to a subsequent login, this will remain in the same position. So it'll be saved for your next session. Next here, we have the participants. And we could actually use the top chord. Actually, no, let me just remove that. And okay, perfect. So as you can see here, we have 361 participants, and this table view provides essentially the information that we're looking for really at a row level. So you have the participant ID, the study code, DB-GAP, the diagnosis associated, phenotypes, and the biospecimens associated. So this particular participant in particular has 135 biospecimens and 8 data files attached to them. So I'll show you all a little bit here what I was discussing about the saved sets. So if I was interested in, let's say, just the first 20 of this table of results, what you could do is, if you want to have more so of a static, and to the question that was asked earlier, where you don't want your save set or your be affected by a data release, you could save it here, and it'll be based on the IDs of the participants. So the filters will not affect the new data release shouldn't affect your result of your participants. So let's just put HTTP participants. And if I save that, you could also add to an existing, so I could go to the next page and I could add them, and I could also remove some of the participants. So if we go back to the dashboard here, you'll see here, similar to the save filters, you could click here and it will redirect you to the data exploration page. You could again rename them, delete the set. And we also have the sets for the biospecimens and files. So this is really more so of a static entity. It will not be adjusted. If there's a new data release, again, it will not vary. The results will be the same. So let's go back here, and you'll see here it added it to the query, and we have the 20 participants here. So let's just close that and come back to our original query. The other thing that we could do here in the tabular view is we could download the clinical data for these participants. So I could select all of them, and essentially by clicking select the selected participants, I'll be generating an Excel sheet with tabular data, and at times we do provide a little bit more detail that could fit within this table. We could also export as TSV. So this export will show exactly what we're seeing here in the table. So if I were to export as TSV, then I would see these specific columns. However, it does adjust if I were to adjust, we do have a column selection tool where we don't show some of them just because it makes the table a little bit more cluttered, but we could add them. So let's see here, we have the race and ethnicity added. We could adjust here the preference of the position of the columns, and it'll adjust it in the table itself. And now that I've added 2 new columns, then I could again do the export as TSV, and it's going to take essentially the image, the data that's shown here. So that covers that here for the participants. Coming to the biospecimens. So this is what we refer to as the virtual biorepository. And what's really powerful here is that the virtual biorepository can be queried across various filters of the participants' data files and, well, the biospecimens as well. So that's really important in terms of if you're looking for a particular, if your research topic, again, is looking on a specific type of diagnosis, if you're looking at congenital heart disease, for example, then you could use the filters on the left-hand side So let's just put that in actually. Let's put congenital heart disease, and we have it here. It will filter essentially the biospecimens according to that condition. So why that becomes useful is that you could search across all of that. And yes, we understand that the whole queryability portion of it, but the point of this virtual biorepository and our vision for it was really to encourage discovery and sharing of samples, really. So what we see here is we have these sample IDs, we have the study, the sample types. So this is really, again, we could see it on the filters on the left-hand side. So you could easily look across. If you're interested in DNA samples, then you can look for DNA samples. So let's actually do that. And so we'll have the parent sample and finally the collection ID. So what this collection ID is that what it does is that it encapsulates all the aliquots that were taken from a specific blood draw. So if I were to add that and just remove my other filters, you'll see that this is the same collection ID, and we'll see that there are all the different aliquots associated to them. And if I open here the column selection bar, we'll have the specific container IDs. And from there, you could decide, well, is this sample available? And you could also, again, search it on the left-hand side. They're currently all available. And if it says yes, well, that's actually something that we're going to be working on. But the point is that we're planning on having a request a biospecimen button here. And essentially what would happen is that if the sample is available, then you could select them. And then you could click the request biospecimen button. And what we'll be providing is the contact information for the institution or the biobank that's storing the sample itself. So that's really a powerful tool, and we're going to be working on that in the near future. The next portion here that I want to address, again, so similar to the participants, we have the Save Biospecimen set. We have the download sample data. So again, it'll be generating a CSV file. We have the export as TSV and the various columns that we have here. So let's jump finally to the last column here. Maybe I'll empty out a few of these queries just to make a little bit more space. Oh, I'm sorry, I forgot to mention something. On the left-hand side, we do see that there's a search by ID, so that could be definitely done. But you could also search by sets. So if I were in the participant sets and I had saved one earlier, you could easily, instead of going back to the dashboard, then you could easily select here the participant sets here, and I'll add it. And again, this is what we had seen earlier in terms of the save set that we had. Another important thing that I forgot to mention about the save sets is that these save sets will actually be used as a basis for our analytical tools in the future. So this will include comparison of cohorts at the clinical level or even at the gene or variant level. We also plan on having enrichment analysis, I guess with geo terms, so gene ontology terms. So really the save sets will be an important piece to all of that, similar to the variant workbench. So let's come back.
While you're on the topic of participants, I would like to address a question that came out in the chat about participant IDs and the potential for participant overlap across cohorts. So each study has study participants IDs, and within the study, there's no duplicates. Okay, so within the HTP, you know, each participant has a unique ID, and the same is true across the other studies. And I can also share that of the studies that you're looking at there's no overlap between the HTP DS360. Would you share the study again so that I can name them? Let me do the reverse. The only study where we have not been able to define overlap is DS Connect, right? So there is all the other studies are non-overlapping. There is a potential that a small fraction, or who knows how small fraction of DS Connect participants may be also in the other 4 studies, right? With that being said, the DS Connect participants don't have any of the omics or biospecimen data sets. So hopefully I'm addressing a unique question that if you are working with the other cohorts, the non-DS Connect cohorts, you're pretty safe in terms of participant overlap. If you are looking at the level of clinical profiles and you are mixing and matching DSConnect with the other 4 cohorts, there is potential for some overlap. I hope that makes sense. And I should point out, we're working on a solution to make sure that if a participant is in 2 studies, we can know who that is and join the data sets coming from 2 studies. That would be an amazing achievement. We're working on it.
Yeah. And just to add a couple comments around that, and thanks so much for the questions. So as Joaquin highlighted, linking across those studies is one component, but especially linking across studies where data are collected in very different manners. So, for example, in DSConnect, you have the participants essentially themselves providing data, versus in a research study, you have the investigator potentially extracting data from the electronic health record or from a survey or some other component. And you can imagine that those 2 different sources may not 100% overlap in time or scope in ways that could be additive and synergistic. And so that's really a very unique context for the Include Data Hub. The other layer, and I think this was implicit, and perhaps JP mentioned it and I missed it, but as you query across either the participant or biospecimen or data file context, especially in the biospecimen side, I just wanted to reference something I mentioned earlier, which is that that iterative cycle of essentially generating one type of data modality and then being able to ask the question, now that DNA sequencing has been generated from this sample, is there more of that sample to generate another type of data modality? That use case of that asynchronous—
Is it a talk I'm listening to right now?
Oh, I'm sorry.
Sorry, I meant to be muted.
Okay, no worries. But that iterative scientific process, which is the way really science is done, where we typically generate additional data modalities, not all at once, but at different time points, and assessing whether or not there's still sufficient material to do that research, potentially by 2 different people, 2 different groups, but combining that data downstream. That type of community-based discovery and work is something we really hope to empower.
Great, good question. So coming back here, just to finalize here and do a segue to the data files. Essentially, the last column here for the biospecimens, as Adam was alluding to, these specimens actually are associated to files. So you could easily click here and you'll be redirected to the data files associated to the biospecimen. So jumping to the data files here, this is really going to be the step right before the analysis portion. So this is where we're going to be explaining how you're going to be copying files into Chromatica and use it downstream for your analyses. So the various categories, filters, sorry, that we have here are, again, portrayed in the table as well. We have the file IDs, the study, the data categories, so whether it's genomic, metabolomic, proteomic, the data types, align reads, and the experimental strategy, whole genome sequencing for this case, the format, and again, the association with the participants and biospecimens. So the one thing that I want to address here is the different data types that we have in the portal. So we have 2 levels at the moment of different types of data access. So we have controlled and registered. Eventually, we're surely going to have open access as well. But the registered level access is essentially if you register and follow, which is what we did during the registration process. So once you're in the portal and you have an account, then you would have access to that registered data. In terms of control data, let's start with this one. Essentially, this is governed by your ERA Commons NIH account that's tied to the dbGaP study-specific access that you have. So let's go back to the dashboard. And so just before that, actually, so this is displayed here. If I had registered data, for example, then this would be displayed as registered. And right before going to the dashboard, sorry, I forget to mention here, we also display a dynamic column for the file authorization. So currently I'm not connected to any platforms. And so it shows that I don't have the authorization. This would be the same for the registered tier as well. So how do I get the authorization for controlled data? In my case, I do have an ERA Commons account. So let's just jump back to the dashboard. And as I was mentioning here, we have the authorized studies widget. And essentially, this is where you're going to be connecting to your ERA Commons account to have access to those controlled data accesses files, sorry. So if I click here, I'll be redirected to the ERA Commons authentication service. And with a successful login, I'll be brought back here to the portal. And as you can see, we have a list of the different controlled, the different studies that have controlled level files. So as you can see here, we have the include loop genomic analysis of congenital heart defects. We have here the authorization of the different files. So at times you could have the authorization for all the files of the study, whereas at other times you could have only a portion of it. So we display this that you have 878 files available, authorized out of 4,083 for the HTTP study, for example. And we also display the accession number here, the dbGaP accession number, and the consent code as well. So let's click here, and we'll see that we'll see the different data files that we have here. So this is more so the genomic level information. So let's just take that off. And in terms, so as you all saw, oops, I shouldn't have taken that off actually. Just give me one quick second. So this was essentially the data set that I was looking at. And as you could all see, this column is in fact dynamic. And now that I'm connected, it shows that the lock is unlocked. So I have authorization to push these files to Chematica. For the registered tier, the way that's governed, however, so let's just take that off. I still have a lock here. So in terms of accessing, although you all must question how come I don't have access, I registered. Well, the only thing that you need is to connect to Chematica, which anyone can register to Chematica and connect to it. You could do it via your ERA Commons, and I'll show this right now. But just to give a little bit of background in terms of what is Chematica, because at times I feel like individuals see the Chematica projects, but they don't know what exactly it is. But in fact, it's a very, very powerful tool. So Chematica is essentially a sharing platform, a cloud-based environment and sharing platform to allow users to analyze data and store data, not only in the Include platform, but across multiple other platforms that touch on genomics or multi-omics. The Jack and Michele that are on the call are going to be discussing how to do various analysis on the different pipelines that are available in the tool. So we'll dive a little bit deeper there. But coming back to the registered tier data, so essentially you need to be able to connect to Chematica to have access to those and copy them in your Chematica project. So here I'll I should have access, it should be working. So again, you could connect through ERA Commons, or you could register as a new user under Chematica. So as you could see, I already have, so this dynamically talks to the Chematica platform and it communicates with it to show me the different projects that I have. So what's in these projects are the files that we actually copy from the data files tab in the data exploration. And I'll do a little demonstration of that as right now, actually. So let's go here to the data files tab and let's let's do the data category proteomic. And so proteomic specifically could be really interesting for clinicians that may be interested in inflammation and immune markers, because the proteomic, the research data set that HP did research on was actually on immune markers. Joaquin, did you want to provide a little bit more detail in terms of the immune markers that you all did?
No, for those aficionados that may be interesting, this is what we call a targeted proteomics by multiplex immunoacids enriched for immune markers of interest to those interested in immune dysregulation and Down syndrome. There is more untargeted proteomics coming in the pipeline with thousands of features. This is heavily enriched, as you will explain, for immune markers.
Thank you. And as you all saw, prior to connecting to Cubatica, I didn't have the authorization, but now I do. So what we're going to do now is attempt to copy these files. Let's say I'm interested in doing a downstream analysis with the tools that are available in Cubatica. So I could click here and select, let's start with selecting 20, and I could click on the Analyze in Cubatica. So here a modal will be opened up, and you'll be able to select the project in particular. So as you all saw, those were the projects that I currently have living in my Cubatica platform. I could also create a new project. So let's write HTP proteomics as the name. You could select your billing group. So mine is already selected. I'll create that project. And as you all see, it has been created directly from the portal, and I could copy those files. And as you can see, we could see that it is a success, and I could verify that by either clicking here on the open project in Cubatica directly. And so that's going to redirect you to the Cubatica, but let's do it from the dashboard. And so if I'm coming back here, we see the HTTP proteomics, and you see that there's one contributor to this, or collaborator more so, for this project, which is myself, but you could also collaborate with other members. So that it could be much more than one. And I'm sure the Cubatica team will be providing a little bit more details in regards to that. So if I click here, you will see that there are the files attributed to the proteomics, and they're all listed here. And as you can see, we have the 20 files that we generated from Include. So it's really a powerful tool. It's an all-in-one feature, and yeah. So that covers it. And again, the save files also, you could also save sets for the files. So jumping back to the presentation, I want to do a little bit of more interactive walkthrough, just to see if everyone was listening in. So let's go and do a few exercises now that we know how to use the tool a little bit more. So let's start off by going to the data exploration page and find how many participants are females with Down syndrome and have hypothyroidism with files of data type protein abundance, absolute protein concentration. So let's go ahead and do that. I'll clear my query and go to, actually, yeah, sure. Let's clear the query. And let's do, so it was sex, female with Down syndrome. So we could do Down syndrome status, and I encourage everyone to follow along. So trisomy 21. And I believe it was hypothyroidism. Let's select that from the diagnosis browser. And as we can see, we have 595 participants with this exact term. Let's apply that. And for the data files, let's jump to the data type, and it was the protein abundance, absolute protein concentration. So now that we filtered across that, the question really was, how many participants have this set of filters? And so we could see that we have 96. The next exercise that we have here is to download the clinical data for males that are Hispanic or Latino with Down syndrome and sleep apnea syndrome. So let's go ahead and do that. We could create a new query. So male, ethnicity, Hispanic or Latino. And sorry, I forgot the conditions. So let's do Down syndrome. And I believe it was sleep apnea, if I'm not mistaken. So as you can see, I could take the more general, the note that's a little higher, but there are also some diagnoses that are a little bit more precise. As you can see here, that drills down. So let's select sleep apnea here. And so if we wanted to do a download clinical data, again, you just select every row level. You select all to have all of them. And you simply click on download clinical data. So that's that for this one. The third question here is find how many samples for participants that have Down syndrome with DNA samples that are available. So again, this is really to show the power of the virtual biorepository. So the question here, let me just reclose all of those. was to find the participants with Down syndrome with samples of DNA. So you go here to the biospecimen category, you select DNA, and we wanted them for those that were available. So we have 680, and now as you can see, we have 680 biospecimens for this specific query. And so again, in the future, we're going to have a request biospecimen data button, which could help through the process of getting those biospecimens in real time, and you could use it in your laboratory. And finally, the last question here is to find and analyze files in Cavatica for files of type align reads for participants that are female with Down syndrome and celiac disease. So let's first start off by, I could have also used some of the queries that we already prepared, but let's go ahead and do this. So let's again go in diagnosis, celiac disease of type aligned reads. And let's do the analysis and push these files into Cavatica. So let's select those. And as you all could see, since I did my connection to the platforms, we see that I have authorization, even though it's a controlled data access level data file. So let's click here and analyze. I could also create a new project, but for the sake of this presentation, let's just put them in the same project here. And you can copy files, and you can see that they are associated to HTTP. So there you go. Hopefully you all followed along those 4 exercises. I may have went a little bit faster since I'm used to the portal, but pardon me for that. And in terms of future work, really, we're planning on in the upcoming months to develop a variant search exploration tool, which is something that we have within the Kids First platform. And essentially that will provide variant and gene information at a participant level. And essentially you'll be able to verify the variant attributed to specific phenotypes or diagnosis, for example, and look at different pathogenicity, the pathogenicity of these variants. And so it's going to be a very useful tool once we have it in the Include portal. We also, again, plan on having more analytical tools, which I was mentioning in the dashboard. So enrichment analysis tools with geoterms and cohort comparison. So it's going to be very useful. So that covers it for mostly the workshop of the portal. So I'll invite anyone that has any feedback or questions to ask away.
Please keep entering your questions here in the chat. We're doing our best to address everything in real time. If not, we'll follow up. But while you guys are writing your questions, I would like to invite Dr. Huiqing Li, a project scientist at NHLBI, who along with many other NIH officers is a strong collaborator in the design and oversight of the Data Hub, to briefly explain to us the Cavatica credits and billing, so that you guys have seen how JP was pushing data to Cavatica and was being asked, about a billing group. Jack, hopefully I'm not stealing your thunder. Jack will be doing more of analysis in Cavatica, but I want to demystify the process of billing here, and maybe Dr. Huiqing Li can spend some time on that.
Thank you, Joaquin. Hello, everyone. My name is Huiqing Li. I'm a product scientist for the Info DCC. So I hope you got excited to hear, what we have been doing here with the DCC. And I just want to let you know that we have a Include Cloud Credit Pilot program. I can present my screen here, so you can find more information through the Include website. Can you see my screen? So if you go to, okay, I can show you. It's not very easy to find it, but there is. If you go to home, about is a health center, and then have documentation here. Okay, Cloud Credit Pilot Program. So if you are interested in using Down syndrome data and to do analysis on the Cladica platform, or if you don't have the data, if you don't want, I mean, you don't have, you're not a data contributor, but you want to use the Down syndrome data through the cloud on the DC Data Hub. And we will offer you initial $1,000 cloud credit. And after that, if you are interested to get more to support your further data analysis, you can request up to 20,000 cloud credit. So in 20,000 means if you have developed any tools to share with the whole communities through Cladica, we will offer you up to 20,000. And there's, if you are interested in this pilot program, please feel free to reach out to me. This is my email address. And I think that's it. Let me know if you have any questions. Yeah, just feel free to reach out to me. Thank you. Okay, Joaquin, I think that's it for the cloud credit.
Thank you, Chin. So then next, we're gonna move to more of an analytical exercise. Sorry, I was monitoring the chat for any questions. So we're gonna hear from Jack DiGiovanna from Seven Bridges, developers of the Cladica platform. He's gonna demonstrate an analysis. Jack, all yours.
Thanks so much. Just sharing my screen a moment. Okay, can you see some slides? I hope.
Yeah, but we're not in, now we're in presenter mode. Thank you.
Okay, so thank you so much. Great to follow everybody's introduction, and JP did such an amazing job that this will be an easier task for me. But really, I wanted to talk about the handoff there from the exploration and discovery in the portal, and then moving on to what further analysis that you want to do. And one of the things that JP left off on was, okay, what about adding an enrichment analysis? What would you do there? So that's what we're going to walk through today. As JP introduced, Cladica was co-developed with the D3B team at Children's Hospital of Philadelphia around 2016, initially focused on pediatric cancer, bringing together these two groups, CBTN and PNOC, and then it became a key platform for Gabriella Miller Kids First, for the INCLUDE Data Coordinating Center, and Common Fund Data Ecosystem. So it's bringing together all of these different types of data types for analysis, and it's bringing together people so they can collaborate, different tools, and with the really phenomenal support of the NIH that Hui Cheng just mentioned, letting people get started and get a feel for the analysis on the cloud, providing more substantial support up to $1,000, and even up to $20,000, depending on the scope of the research question that they're trying to solve or the tool that they're trying to build. There's a number of different analysis capabilities. I'm going to focus on this one here in the middle. I don't want to drag out the demo, so trying to show everything, but thinking of production pipelines and scaling. So this is really, I have some data, I want to configure a tool, press a button, and then just get the results. This could be differential expression, which we'll show later. Or it could be, really you're taking all these samples, you're realigning, you're joint calling, going across entire cohorts. And I think these capabilities are important across both time and collaborators. If you go back to the spectrum slide that Adam showed in the beginning, different users are going to use these different tools, and you're going to use these tools at different times. You might go earlier in time or to a different type of collaborator and user and want to do prototyping and interactive analysis. So you're not sure what you want to visualize yet. You want to do some statistical analysis. You want to change the algorithms. Well, you could do that in Python or RStudio. Once you've reached a good point with that, then you can harden that and push that back into the production pipelines. And finally, what we're really excited about in our INCLUDE Data Hub is being able to develop custom web apps. So now you're getting away from the more technically savvy user and analyst, and you want to share your results with someone who's more scientifically savvy. and they can interact really well with GUIs. They don't really want to be writing bash scripts. So being able to work within the same secure and compliant environment with the same protections on data access and costs, but organizing your results into a shiny dashboard, into a shiny app so that people can interact with it. The demo today, we're going to start, following up really nicely on JP's demo, start with gene expression data, which was HTTP data that was processed with Callisto. And we're going to leverage Cavatica for further analysis. That'll be 2 steps. So using DESeq2 for differential expression, and then gene set enrichment analysis. So with that, I'm going to switch to the portal. This is, I'm sorry, I just have to move these videos out of the way because they're jumping over my portal. This is my portal. It looks different than JP's. But it has the same flow. It has the things I'm connected to in Cavatica. And I want to explore data. So I'm going to go in directly to the data exploration tab. My filter is empty because I cleared it out. And I want to start to look into what I'm going to bring in. So I'll start with the study code and go to HTTP. And then I want to look at the types of files and grab my sample type. I'm sorry, sample type. Biospecimen, sorry, jump down one. My sample type in the biospecimen will be RNA. And my data files, I'm going to take tab separated values. So this has gotten me to 400, and I have the summary overview. I have 411 participants from 385 biospecimens and about 1,900 data files. And this is a mixture of different things. I can select one or actually select all of the results. I also am authorized. This is data that I'm authorized to use. So I can directly connect it over to Cavatica. So here we're going to push 1,900 files over. I'm going to put it in, I made a project earlier, live demos are good, and copy all of those files in. And I've already logged in, so that's why it's not asking me to log in, but it asks JP to log in. And I have Cavatica open in the next tab. And it gave me an error, surprisingly. So I have this demos are good file. Just want to double check that I can copy that over. And so, and when we say copying here, I want to highlight that it's not actually moving the data. It's actually just creating a pointer to where the data lives. This is really important for, the efficiency of the storage and the speed of the transfer, so that we're not making, with every single user, we're making an additional copy of the data. Additionally, it's important for controlled access and authorization, so that anytime someone tries to access the data, so there we go, it copied over, and I can open the project in Cavatica. And here I'm in the project. I'm in the first page. And as JP showed in the beginning, there's also a help function here. It's going to give me context-aware support that says, hey, you're in the beginning of a project. Do you want to add collaborators? Do you want to leave? What do you want to do? Or you can send in a help ticket to our support, which will capture the system state and they'll get back to you. But those files that copied, here we can see the project. We have that many files in there. There are DIRS links here, which is what I was saying as a transition there, that we're not actually making a copy, but we're only pointing to the data so that we don't have to store an extra copy. And so that if it's controlled data, each time I want to use it, or if I add a collaborator to the project, we're making sure that he or she has access to that file and cannot get to it without access. So now I have the files, and remembering along our demo, we want to do differential expression. So I need to have an app to analyze that. I don't have any apps in my project because it's new. So here I can go to the app gallery on 7Bridges. And this is just loading up. And oh, sorry, I wanted to follow a different path. So I can go to public apps. It just gives me a slightly nicer overview. It's the same apps that we're seeing in both cases, but you have this little showcase featured apps. So here is GATK, which was actually developed by the Kids First DRC. We have things that were developed by the Plan Lab, by D3B. So it's both tools that have been wrapped and optimized by Seven Bridges and also by users and collaborators and partners. But you can also browse through and search over different types, categories, or toolkits. In this case, there's many of them. We wanted to look into differential expression. Here you can see Callisto, RNA-seq, cufflinks, but I think DESeq2 is the one that we want. When I open that up, or when I open any app up, you get a description of what it's doing, what license is it under, who's developed it, which version, links to documentation. We also try to have for every app some of the common use cases, how you would call it, if we introduce any changes, and some benchmarking. This is particularly fast, so looking at 3 samples, it should cost less than a penny. But just so that we have that and understanding what are the inputs, settings, and outputs. So here I can see this looks like exactly what I want, and it has a set of inputs that it takes in terms of it takes TSVs. So I want to copy this app into my project. However, there's a problem. So when I go back to the project and I go back to the files, we wanted to look at particular abundances. So I can filter this in a number of ways. Let's say abundance. All of the TSVs that I have are actually compressed. They're all tsv.gzs, and that application doesn't take a compressed file. But that's okay. I can just quickly grab another file that does that. So decompressor. Bring that in and run that in my app. So this is a very simple app. All of it's doing is, imagine just unzip. You have input files in the middle. On the left, excuse me, in the middle is always the configuration for any app. So this is just whether or not the output should be flattened or put into a folder. We'll leave this as true because it's single files in. And here we can see we want to grab the tsv.gzs, and we can select all of them here. We can take the first 100 of them, save that selection. And it has a helpful validation check here as a user. Okay, I'm trying to run this app, and it's warning me, hey, Jack, this app only takes a single input and you're giving me 100. What are you doing? So that's not what we want to do. We can change this so that it does batching rather than running trying to put them all together. So as soon as I do that, I'm sorry, and then I have to pick go by file, and it will create one task for each file. You might imagine that you do other types of analysis and you want to leverage the metadata that's associated with the file. So you could use instead file metadata. You can imagine if you were doing trios, you might want to do family ID and whatever you were using for your analysis, you could use, and the executor would be able to use that to break up the execution into different groups of that size and run it. So we can run these quickly. It warns me I'm about to run 100 tasks. So no problem. Go ahead and fire that up. So I come back to the dashboard here. You can see that starting to run. And if I open it up, it'll see that it's actually putting them all together. Some of them here are queued because it wants to make sure that I don't come on and just fire off thousands and thousands of tasks to make sure that there's balanced support for all of the Include users. But this is a very short job that will run quite quickly. And as they finish, we'll be able to do further analysis. Then when we want to set up, each particular one that's running, you could look in and see what's going on. You could get into if you wanted to debug what's happening, getting into the stats and logs, seeing what's happening. Actually, this is just moving all the files, so there's nothing to look at the moment. Then the next thing we want to do is set up the task for set up the DE-seq tool. Once those are all done, we're going to want to do differential expression. So I'll set this up to run already. You can see here, unlike the decompressor, this is a much more complicated application. So it takes more inputs. It has more settings. I'm going to take a gene annotation file. I can pull this from public files. I know that this takes a GTF. It takes a reference file. The type of file is GTF. And I see there's this GRC CH38 ensemble here. There's actually a newer one that I want, so I could go instead to one of my other projects. So I can go pull this from one of my other projects instead of that. From the data demo, take the extension GTF and pull in this one. It's going to copy it over to the project. I'm going to use as a covariant of interest sample type. The analysis title, I'm going to use T21, D21. And you can see here it's highlighting in red the inputs that the configurations that absolutely have to be given or the tool will fail. Other ones that can be left at default are left in gray. So if you remember in the intro, this was with Callisto, and I'm going to ignore this version. So I'm starting to set this up, but I don't have my input files yet. So I'll move away from this a moment and see how the other tasks have gone. 65 of these have already finished the decompression tasks. When a job is completed, so we had the input file, the configuration, and the output. If I click on the output here, excuse me, I can see that any metadata that's associated, so this metadata came from the portal, and it propagated through. Oh, so the FHIR document reference came through here, and we're able to see the TSV file, the script that made it. We can have a preview of it. It's a table as you would expect. So now I'm going to come back to the task that we just started to make, the differential expression, and bring in expression data. Another way I could filter rather than on type is I could instead start to look for, so I need TSVs because that's the type that it takes. And I will use sample type because that's what I said the covariate would be. You can see in the table here, the D21 versus T21. So bringing these in. And now I have 11 files that I could bring in, select them, and have that as my input. And so we're all set up now. We have the expression data that we want to look at, the covariate of interest, gene annotation, set this up to run, and this will take that data. I didn't run all 100 samples because in the interest of time, but I could have as easily put 100 samples into this or 400 samples as these 11 that was put in. So while this is running, some of the other aspects of the project would be, okay, now this is great that I'm running this, but I'm not going to know how to analyze the results. So I want to invite people to the project to collaborate with. So I want to bring in Joaquin and Adam. And I want them to be able to look at what happens. They don't need to execute any data, but I just want them to be able to see what we have. I want to bring in Michele to try to make this more interoperable with other data sets. And I want to bring in Nirvana because she made the tools that actually allowed me to do this demo. So happy for her to have all these different controls. So just that quickly, you could have different role-based access control, allowing people to have different levels of authority within the project. Also, what results they could see, what tools they could make. So it's a second wrapper around, ultimately, if they try to access any controlled data, that's going to go back for authorization, and they will be checking with their dbGaP permissions. But for derived files or for uncontrolled files, they would be able to see them in this project. The other.
So Jack,
yes.
Sorry to interrupt, because I cannot help but to pause us here to share with you how spectacular is what you're doing now. Not everybody in the audience may be familiar with transcriptome RNA-seq analysis, and what you are doing here in real time, which requires some knowledge of Kavatica and finding the apps and so on and so forth, is a very sophisticated analysis, of thousands of messenger RNAs in the entire blood of people with Down syndrome versus people without Down syndrome. Something that many in the call will agree requires very educated bioinformaticians and biostatisticians that will have to be hired, trained, and paid for to be able to do this. And you are demonstrated here in the labs of 15, 20 minutes, and you are about to show us the result. This is truly spectacular that the Kavatica platform can do this in real time for somebody who just needs to learn a little bit how to navigate themselves across the platform. So I just wanted to pause and celebrate, how outstanding this is.
Thanks so much, Joaquin. Yeah, that's what I wanted to say with Nevenya. She is a senior bioinformatician and was able to configure all these tools and bring them together so that someone like me can come in and just click buttons and just see the results. But it's, as Joaquin is saying, it does take quite a bit of knowledge and expertise. And that's one of the reasons why it's good to have tools that are already wrapped and optimized by those people, and they can be shared with the community to be able to bring that analysis to more and more researchers, right? Not everybody has to do everything. We're all a team together, and you can have your very skilled tool developers, your very skilled scientists building on each other's strengths. So this is just running 3 minutes. It takes about 6 minutes to finish. So we already have a pre-baked cooking show style project where we could see the results. Here you can see Michele created and this really nice project. We have a explanation in the left of what was on, links to the results. So I can look at that DESeq2 run and look at the HTML report that comes out. So you have the same definitions, the same inputs. And now you have, scrolling up a little bit, the experimental design. This one was actually smaller. This only had 5 samples of each from each group, but you can see principal component 1 pretty well accounts for the separation between T21 and D21. There is still some overlap, and PC2, there's also some clustering there, unclear what that would mean. You can see sample to sample distances, raw and normalized counts, and looking at the dispersion. And you know all the plots that you would expect to get out of this type of analysis. You could go further than that and go into gene set enrichment. So I go back to this project, the tasks that have run. So I took the output of that differential expression and put that into gene set enrichment workflow. And now you have that CSV output, use this set of gene sets, and as a overview, you have a similar report showing that 48 of 50 gene sets are upregulated, the different significance. This is all linking out to a lot of different results. So it'd be more interesting to look at what each result was. I'm going to highlight just 2. Coming back. It's all in a zip file here. You have your up and down regulated gene sets. So for upregulated, there's a number of gene sets that are upregulated. For down, there's less. So I'll go back to the overall files and we'll look at the interferon gamma results. So here I need to pull up the PNG. And this is the hallmark plot interferon gamma response. And you can see here the enrichment, the rank enrichment score. And you can actually get into a deeper analysis of that with the HTML. Now looking at specific symbols where they rank in the gene set, whether or not they are positive for enrichment, which most of
Jack,
these are.
Will you pause for a minute? Another moment of celebration. Will you hit on that thing called MX1? And would it show any data if you hit on it?
So MX1, it brings us to the ensemble here, and
Right.
you can
So
see.
What you are demonstrating now, you just run, a transcriptome analysis of the whole blood of people with Down syndrome, 10 of them or so versus 10 or so controls, and you had a strong signature of interferon gamma. You also have another one called interferon alpha. This is something that we and others in the field are studying. And the top gene in that signature is this gene called MX1, which is an interferon-inducible gene, which we use to fight the flu. This is an antiviral protein. And it also happens to be encoded on chromosome 21. I just wanted to pause there for a moment to you're getting real data, real discoveries. Sure, interferon hyperactivity in Down syndrome has been documented before, but you're now rediscovering that or reproducing that in real time here with the data in the data hub.
Yeah, thank you so much for that. And yeah, all in all, even though we had a pre-baked cooking show here for the sake of everybody's time in the demo, running these all back to back in under 15 minutes, you've gotten the data from the portal into Cavatica, set up your tools and run them, and gotten to these results. And the only other thing that I wanted to add here, I suspect the other project is probably finished now too. Yeah, this one is completed too. So we could see having more samples, how would that change the results in terms of the principal component analysis? This is really working a lot. So you get a little bit clearer separation here along PC1 between the two groups, but there is still overlap. We focused in this demo on taking data from HTP, from the portal, bringing that into Cavatica and running that in the analysis. One thing that I don't want to gloss over is that this is an open analysis system. So you can also bring in your own tools, and you can also add your own files. So you may have the gene expression that was in HTP and you want to bring in your own data there. So you could imagine doing that from a GUI-based uploader, if you have them in a manifest, if you have them as DIRS IDs. So I don't know whether folks in the audience are familiar or not familiar with this, but the take-home messages here is you have a lot of different mechanisms to bring in your own data to co-analyze with the data sets that are in the Include Data Hub, and also other NIH data sets that are available. Maybe you want to bring in, assuming you have authorization, of course, but if you're authorized to bring in TopMed data, to bring in Kids First data, other data sets that you want to co-analyze. So bringing the tools together, allowing you to collaborate with other scientists and researchers, and bringing data sets together so that you can answer your research question. I can go on for hours, but I want to pause there if there's any questions or anything that people want to particularly see.
Thank you, Jack. Again, truly spectacular. This is not a trivial analysis that you just did in the past 15 minutes, which usually would take a very well-educated, well-trained bioinformatician and pre-packaged applications and pathways. Here, it can be done in 15 minutes. What you just basically did was identify messenger RNAs elevated in the blood of people with Down syndrome. So we're doing great on time. Well, we're doing all right on time. It's 11:45. We have another 15 minutes on the clock. The chat has been quiet now for a while. So I think we can afford to take more questions and comments. And if it wasn't obvious to our guest, because we didn't say it, you now have access to 55 terabytes of unpublished data, right? You just have access, let me say that again, 55 terabytes of unpublished data. talk about open science in the INCLUDE project. So the 5 studies currently in the Data Hub are leading by example. The EXO studies, Lupo, Sherman, HTTP, the HTTP study, DS Connect, leading by example, taking unpublished data, releasing it ahead of publication. Of course, these various teams are working on making discoveries out of these data sets, but it is truly a new age in Down syndrome research where the data is getting published ahead of the first discovery so that we can all get busy and start analyzing. And please be prepared for in the future for us to work as a community of data analysts. We're going to have seminars, include Data Analysis Club, another such exercise to work collaboratively in the analysis of these data sets. But I'm also hoping that by leading by example, we can instigate data sharing ahead of publication, participation in the EXO-1 funding mechanisms, participation in the R03 data analysis mechanisms. Is really a phenomenal time for people with Down syndrome with this research where scientists from all disciplines can come together to analyze these data sets, most of which have been paid with taxpayers' money. And I will pause there, monitor the chat. It's been quiet. Thank you, Amy, for the note. And yeah, we have 13 minutes on the clock. I wanna thank my colleagues. I thought it was a great demo, and we can take any questions or comments now. Also invite my colleagues from NIH if they wanna use this opportunity in front of the community to share any of the information about the INCLUDE program.
Thank you, Joaquin. I just posted the 2 opportunities, EXO-1 and the R03. Please feel free to reach out to me for any questions you have regarding to the EXO-1 or the R03. Thank you. I forgot to mention that, for every user, you can apply for $100 just to get familiar with the cloud to do a test on your end on Kavatica if you are interested. For anyone, not only researchers. Yeah, and the $1,000 is for a research project.
Thank you.
Yeah, I think Surya shared the link in the chat. It's a really lightweight sign up. It's maybe 3 or 4 minutes to go through that email and password or link your ERA comments. So we could share the link if you're interested. And if you have any questions, please contact support and we'll get you on the right path.
So we have a question from Jenny. Hi, Jenny. Lawrence, good to see you here. Right now, Jenny, we have only one RNA-seq data type, and that is whole blood transcriptome, but there are other transcriptomes coming online. And as we have samples from other cell sources, we'll make sure to indicate that. So this is the beginning of the journey, and you will see there as you continue to sign in into the Data Hub that we're going to have T-cell transcriptomes, B-cell transcriptomes, white blood cell transcriptomes. There are hundreds of white blood cell transcriptomes in the EXO-1 pipeline, and you'll be able to select for those. The question about age was also addressed earlier. There's going to be age at specimen collection, age at clinical data harmonization will be representing age in all the possible ways that it can be used. And I think differences in normalization between data sets, you will have access to raw data, right? So you can, employ your favorite RNA-seq transcriptome normalization tools. But your question perhaps is related to Bernard's question. Thank you, Bernard, for coming today. Bernard Core from the Benaroya Institute, about batch effects. And, they are probably a non-issue for genomes, less than an issue for transcriptomes if you are using the same sample sort, but there's more of an issue for other of the data types. that we be presenting here, like metabolomics or targeted proteomics, what have you. There are batch correction tools. We'll be sure to educate our users, on how to do proper batch correction when you're doing across different studies. But also, Jenny, within the same study, there may be batches, right? The HTP, for example, has been collecting samples all over the country, and you could say, well, when we went to that conference, that should be a batch. When we went to that other conference, that should be another batch. So it all comes down to our colleagues at the data management core having very rich metadata on, the instrument that was used, if they were not all collected the same way. When we say, plasma, what do we mean? Is that EDTA plasma or citrate plasma or what? And this is where these questions that you are putting here in the chat, this is what tells us what we need to do better, right? We say, hey, you say EDTA, but you're not, there are 3 different ways of doing EDTA. Great, we need to go back and capture that metadata, present it for you because you asked for it. So this is an evolutionary process. It's the beginning of the journey. We didn't want perfect to get in the way of getting you something to start, right? So we will follow our user community, our user base in how to evolve the research, how to collect more data, how to collect more metadata, how to develop more apps, more tutorials. Related to Julie's question, this is our first video tutorial. This is being recorded. You have access to it. If you want to come back to it, Julie, Jenny, anybody, you can come back and watch the entire episode again. Jenny, I don't know if you had any other questions. I see you here on video. Thank you for not leaving me alone here on sharing video. You can tell I haven't shaved in a few days in anticipation for this demo. Great. I want to reiterate once again for those of you who join in later that we have a great training event coming up, the DS3 training event, Data Science for Diverse Scholars in Down Syndrome Research, where we will teach not only everything that you saw today, but then some. I mean, it's going to be a wonderful, immersive, intensive, hands-on workshop focused on graduate students and postdoctoral fellows from underrepresented minorities, July 11 to 22. Many of the colleagues on the call today will be there as instructors. We will not only use the Data Hub and Kabatica, but we'll do a lot of other stuff. Not just data science, we're also going to be providing training on professional skills more broadly defined, including how to write a good specifications page, how to put together a strong NIH bio sketch. We're going to have networking with invited speakers, and which reminds me that you may be hearing from us. Many of our colleagues on the call may be hearing from the Include Data Coordinating Center about inviting you as a guest speaker to the DS3 training event. Great. Well, if no more questions or comments, I want to thank again my colleagues at the Data Management Core and the Data Portal Core, and also our NIH for a great demo today. Again, the beginning of a journey, and be on the lookout for more updates from us for additional educational opportunities. We'll be at the T21 Research Society meeting this coming June. Looking forward to connecting in person with many of you. Thank you very much, and I'll give you back 6 minutes on the hour here.